Okay, so today I wanted to make a video that goes over the genetics that drive cancer. So cancer is essentially when your cells divide like they normally do, but instead of regulated amounts of division, they go absolutely crazy. And you end up with this large mass of cells that shouldn't be there. And this is a tumor. And this is what cancer is. The challenge around cancer is that this tumor is comprised of your cells, so your immune system, it can't fight it as well. And the drugs that we normally would use to kill these cells, they'd likely also kill the cells that make you. They'd essentially kill you, a lot of these therapies. That's why it's so tough to treat. So the key issue with cancer is too many cell divisions. As we see here, a cell dividing into two, and it can balloon and keep going. Usually the cell division is regulated by something called the cell cycle, which culminates in mitosis, which is the actual splitting and dividing. So as the cell cycle progresses, the cell gets signals from itself to, keep growing, double the DNA, because when you double cells, you have to double DNA. And then ultimately, we can go through mitosis. There are genes that can stop this cycle. They can stop cell division. And these are called tumor suppressor genes. And they exist at these checkpoints in this cycle. So if something's wrong right here, say that something's wrong with the cell DNA, it'll trigger these tumor suppressor genes to wipe out this cell. And it'll go into something called apoptosis, and the cell essentially shrivels up and it dies. So that's what usually happens. However, say that you're an individual that loses these tumor suppressor genes, see that I erase right here. Now this cycle is free to keep going as many times as it wants. So we've lost tumor suppressors, and now the cell cycle can go without any trouble. A second thing does need to happen, not necessarily in any order. There are genes that are responsible for triggering this cell growth. Those are called oncogenes. When these increase, or they get a mutation that causes their activity to balloon, this cycle really starts going out of control, and it'll keep going indefinitely. And that's where you get a giant, massive tumor that is dividing without stopping. The checkpoints mean nothing, and that's because you've lost the tumor suppressor genes that usually stop the cell cycle at these checkpoints, and you've gained oncogenes that are signaling to these cells to divide. So in a given tumor, how we used to attack this was we would send in, these are goofy looking things, sorry. We would send in a mix of chemicals, and let's call this chemo. We would send in chemo, and it would go disrupt the mitotic cycle, and it would stop that cell cycle from happening. And it usually would get rid of a lot of cells. The problem is, chemo really hurts people. It still hurts the good cells too. So the non-cancerous cells, they also get killed by chemo. That's why it's an imperfect way to treat this disease, but it is a really good tool for a lot of patients. So a lot of the times, so say this is a big cell, there are signaling mechanisms on the top of cells that cause growth, cell survival, and a lot of the times they're triggered by certain downstream proteins. So instead of using chemo to just nuke all of our cells, we can actually design small inhibitors, small chemical inhibitors, let's use I, that can just block the oncogenes. So right here is a growth kinase, or a growth gene. We can design chemicals that bind to this and wipe it out. And thus we also wipe out growth and survival all at once. Therapies like this are good because your normal cells aren't expressing these bad growth genes for the most part. Now, the reason that all therapies don't work across the board for cancers is that it's found in lymphoma, is that, remember, the tumors are comprised of, billions of cells, so many. And normally, these cells do share a lot of common genetics. So the problem arises is that, and especially in lymphoma, the disease that I study in blood cancers, some cells are inherently stronger than others, you could say, and they have certain growth genes that make them more, I guess, better, like hardier, better to survive than their counterparts. And that when you use a treatment that goes into the tumor, it can kill massive amounts of the tumor. Say this treatment is very specific for something. However, a lot of the times what happens is that this treatment will leave these cells that we talked about, the ones that were stronger, the ones that could defy the treatment and they could keep surviving. And the problem now is that they're the only ones left in the population. And now the tumor is comprised of all these bad cells in red. Now when you try to apply the same treatment, same medicine, it doesn't work. These cells are all resistant, and that's because they came from the population that was originally treated with this one, but they erased all those old cells, and now only the big, bad, worse cells are left. And that is what tumor evolution is, and that's some of the challenges. It represents a large challenge for us as cancer researchers to find therapies that can stop the cell cycle. There are certain treatments now that can reinvigorate tumor suppressor genes, and they can stop the cell cycle, remember? And then for those oncogenes, we have certain chemical inhibitors that can go and block them. And that's what we need to do to treat a lot of these cancers. Now, cancers are broad. There's all kinds of different ones. So every single cancer, and there are thousands, demands a different treatment profile, a different set of circumstances. Every patient's different, and that's the challenge that's ahead of us. But we're definitely hopeful, and we think this is going to be a really good next couple of decades for medicine. Thank you.